Okay, so we're in the Unit 3 cell unit for the Biology Keystone review. Very quickly, I'm going to go through some of the reading that's in this unit. So the first part of the reading starts on page 25 of your reading packet, and the first thing that we're looking at here is the cell theory. The cell theory is a set of scientific statements that serves as one of the foundations of biology. So it's a unifying theme in biology, and it states that the cell is the basic building block of all living things. Organisms are always made up of one or more cells. All cells come from pre-existing cells, which goes back to all living things reproduce. Cells carry genetic material, which is DNA being the universal genetic code, and all cells are essentially the same in their chemical composition, and all cells need energy to undergo its metabolic processes. So make sure that you take note of the cell theory on page 25. All right, talks a little bit about the purposes of the cell. Let's see if I can arrange that. Okay, page 27 shows us a generalized eukaryotic cell. I'm sorry, generalized prokaryotic cell, which we know is always a bacteria. It doesn't have a lot going on there. It's basically a cell membrane, because all organisms have a cell membrane, cytoplasm, and then a little bit of information going on in the inside. It has some ribosomes. It has a capsid, which holds its genetic information. Some prokaryotic cells have a flagella, which is a tail that helps them move. Some of them have cilia, which are little hairs, sometimes called pili, cilia or pili. All right, you know that your eukaryotic cells are cells that have a nuclei that's separated from the membrane. They're larger and more complex than prokaryotic cells, and we already really talked about this in Unit 1, so you can always go back and look at the Venn diagram in Unit 1, but you can see your typical animal cell and your typical bacterial cell. You can see how they contrast in terms of size. The eukaryotic cells are much larger, and the prokaryotic cells are always small, and they're always bacteria. Another good tool to kind of look over is page 29, because again it shows you some of the different characteristics between prokaryotes and eukaryotes. So make sure you're reading through all of this and highlighting everything, taking notes. It's a really, really good review. Good review of the information that we haven't talked about since the beginning of the year. Unit 3 reading also talks about the different levels of organization. There's that kid, he's an organism, but he's made up of atoms, which are the smallest unit of matter. Smallest units of matter. Atoms come together to form molecules. Our molecules make our macromolecules, which remember are our carbon-based macromolecules, our proteins, lipids, carbohydrates, and nucleic acids. Groups of macromolecules together make organelles. Organelles can be found inside of cells. Cells group together to form tissues. Tissues form organs. Organs form organ systems. And that leads us to our whole organism, that baby right there. So these pages explain a little bit about that. Page 32 talks about cell specialization, meaning that each cell has a different job to do. So in other words, bone cells do bone cell things, muscle cells do muscle cell things, etc. All right, taking a look at page 18 in your overview. It says draw and label the parts. I'm really great with the camera. You guys are so lucky. I'm so good at this. All right, draw and label the parts of a generalized eukaryotic animal cell and plant cell. The answer for that is going to be on page... Come back up again. On page 33 of the reading packet. So a generalized plant cell is going to be a little bit more rigid, a little bit more angular in nature, because it has a strong, rigid cell wall. A plant cell also has a chloroplast and a large central vacuole. An animal cell is a little bit more amorphous in shape. It does not have a cell wall. It does not have chloroplasts. It does have vesicles, lysosomes, and centrioles, which help it during cell division. So those are general pictures of your plant cell and animal cell. Number 1 on page 18 asks us to describe in a Venn diagram the differences between plant and animal cells, and lucky for you, the answer is actually right there on page 34. Animal cells have lysosomes and centrioles, plant cells have chloroplasts and cell walls, and then they all have a cell membrane, nucleus, mitochondria, ribosomes, endoplasmic reticulum, and we could go on. They all have cytoplasm, they all have genetic information, but that's just a general idea of some of the differences between plant cells and animal cells. Moving on to, oh, and then I'm also going to freeze on this picture too. This is from our lab, but it's a really good picture of a plant cell where you can see large central vacuole, nice rigid cell wall, chloroplasts, versus an animal cell, a little bit more amorphous in shape because it doesn't have the cell wall. It's got a cell membrane, centrioles which help in cell division. Alright, so plant cell versus animal cell. The next page of your review packet is page 19, and it says create a T diagram describing passive transport and active transport. Draw a picture of the cell membrane with your illustration. So, lucky for us, we did this already during one of our study guide videos. Alright, so in passive transport, you'll see that there is no energy requirement. Passive transport includes diffusion, osmosis, and facilitated diffusion. Now remember, diffusion is the movement of molecules from an area of high concentration to low concentration until evenly distributed. Facilitated diffusion, even though it's a bigger, fancier word, facilitated diffusion is a similar thing, but instead of just diffusing easily across the cell membrane, it's going to go across the protein channels. So it's using the protein channel to get a larger molecule across the cell membrane, but still without using energy. And remember that the word facilitated simply means to help. So a protein channel is going to help diffuse materials across the cell membrane. And then osmosis is a special type of passive transport that only involves moving water across the cell membrane. Active transport does require energy, and remember that that energy is coming in the form of ATP, adenosine triphosphate. So active transport is going to be moving molecules against the concentration gradient. I want to just take a second to go over the concentration gradient with you and remind you guys what that means. Concentration gradient means that the steeper the concentration, the faster the rate of diffusion. So if you have a high concentration on one side of the membrane, it's going to diffuse very quickly, and that's like going down a steep slide. But if you have a low concentration gradient where you've got like a medium concentration of molecules and then a low to medium concentration of molecules, the diffusion is going to take place a little bit slower, but it's still going in the same direction. Now going against the concentration gradient is like moving this way, moving this direction, where you're trying to move molecules from an area of low concentration to high concentration. So it's very difficult and requires energy. It would be like trying to crawl back up the slide. More energy is needed, more difficulty than just sliding down the slide. So moving against the concentration gradient is like trying to crawl back up the slide. So that's what we're talking about in active transport. It requires energy because in most cases it's moving molecules against a concentration gradient. Our types of active transport, I'm going to zoom in a little bit, are endocytosis, exocytosis, and our sodium-potassium pump. Endocytosis, remember EN, think of the word enter. This is where a large molecule is going to be outside of the cell. The vesicle is going to fuse with the cell membrane, the cell membrane is going to engulf the molecule like Pac-Man. It's going to engulf the molecule, and then it's going to refuse. The cell membrane is going to close again, and so your vesicle is now inside. That's endocytosis. Phagocytosis is just a special type of endocytosis that involves eating something. So amoebas and other unicellular organisms that need to eat their molecules, they do that by undergoing phagocytosis, where they just kind of engulf the other organism, and that's how they eat it. Exocytosis, when you see the EX, just think exit, and this is where we're going to be having a large molecule push its way out of the cell membrane by applying pressure to the cell membrane. The cell membrane is going to break apart and kind of fuse with the vesicle, and then it's going to reseal, and the vesicle will be on the outside. So a large vesicle leaves the cell, moving against the concentration gradient using active transport, by using exocytosis. And then your sodium-potassium pump is a form of active transport where you're pumping either sodium or potassium against the concentration gradient. So you're moving it in a direction opposite the flow of concentration. So those are your three types of active transport, and that was your passive transport. So that was page 19. Page 20 is where we start looking at some of our Keystone review questions. So, all right. So Keystone sample question. Which statement best describes how channel and carrier proteins in the plasma membrane are similar? So here's my plasma membrane, or my cell membrane, phospholipid bilayer, and I drew a few integral proteins in there because that's what it's talking about. Now remember, we always use our scrap paper when we are completing our Keystone exam because it helps us think through the question. So before I even looked at my choices, I went ahead and I drew a cell membrane, or a plasma membrane, and I drew some protein channels in there, and I started thinking about what the protein channels can do. They can be involved in facilitated diffusion, or they can be involved in active transport with the sodium-potassium pump. So those are two things that I remember. So my choices now are that both the channel and carrier proteins allow for the flow of molecules into and out of a cell. That could be right, but I'm going to check all my choices here. Both directly connect the membrane to the nucleus. That's not a thing. C, both use energy to continuously move water molecules out of a cell. I know that water molecules are directly related to osmosis, which is passive transport, which does not use energy. So C cannot be correct. And then letter D is both cause the phospholipid bilayer to form a rigid membrane for a cell. And I know that the cell membrane is flexible, so it can't be D. So for number 10, I'm going to choose A, which is the correct answer. And 60% of the people in Pennsylvania got that answer correct. So good job, Pamela. All right, looking at this picture here, it reminds me of this picture here. Basically the same picture. So let's see what it's asking us. Use the diagram below to answer the question. What is best modeled by the diagram? Is it osmosis, exocytosis, passive transport, or facilitated diffusion? This is an easy one for us because something is exiting the cell by fusing a vesicle to the cell membrane. That's definitely exocytosis. And if we look, I find this confusing that only 49% of people in Pennsylvania got that one correct, but I feel like that's going to be easy for you guys. All right. The next two multiple choice questions had to do with this diagram here. I took some notes on the bottom as I was reading through this. I thought it was important to note to myself that GSGs repair damage to the DNA, which is always found in the nucleus. And I took note that mutations decrease the ability to fight harmful growth. All right, looking at molecule X, it's large and it damages the DNA, which is only found in the nucleus, and molecule Y is large and it slows down or decreases enzyme activity. So I'm just going to keep these notes to my side while I go to look and answer these questions. In order for medicine X to produce the effects that researchers observed, it must have entered which part of the targeted cell? So looking again at molecule X, it damages the DNA, which is found in the nucleus. It has to be the nucleus because DNA isn't found in the ribosome, the Golgi, or the endoplasmic reticulum. So, looking at the answer, we were correct by choosing A. This question is actually really easy. There's just a lot of reading and big words designed to throw you off. But take your time, make notes, and look through this, and you'll be like, man, that's actually really easy. Given the properties of medicine Y, which mechanism is most likely responsible for its entry into the cell? It can't be osmosis because it said that it's a large molecule, and osmosis only deals with water. It could be exocytosis because it's exiting. It couldn't, it could be, I'm sorry, it can't be exocytosis because it's talking about a large molecule entering the cell. So it can't be exocytosis because that's an exit. It could be active transport because it's a large molecule. And it can't be simple diffusion because simple diffusion doesn't use large molecules. So the only possible answer here would be C. The movement of large molecules into a cell requires energy.